Hello, this is Gina Piscatelli with the last lecture for week 1 of ANP2, and we're going to talk about blood groups, blood typing, and transfusion reactions. So I like this cartoon. There's 2 mosquitoes, which, as you know, are parasitic. They want to suck your blood like a vampire, right? And one mosquito is saying, I agree, O positive is rather nice, but my favorite by far is AB negative. Just for a little humor. We're going to look at what's the difference between O positive and AB negative. Not in taste, though, of course. Okay. So blood typing is based on red blood cell antigens. So on the surface of erythrocytes are various molecules, lots of different molecules, and they're called antigens because they're on the surface of the cell membrane. And there are 2 groups of antigens used most routinely to type blood. One group of antigens is called the ABO group. Another group of antigens is called Rh factor. When we say positive or negative, we're referring to the presence or absence of Rh factor. When we say type A or B or O blood, that refers to the presence or absence of these ABO antigens. So we're going to look at the specific ones now. So first, the ABO group. Let's talk about those antigens. These antigens are glycolipids, and there's 2 of them. A antigens, I don't know why they're called A, sorry, and B antigens. Both of them are synthesized by a certain enzyme that is encoded on chromosome 9. Not that you have to know that. So type A blood has red blood cells with the glycolipid A on there. So here's a picture of a type A erythrocyte with A antigens. Type B blood has glycolipid B on its surface. So here's a picture of an erythrocyte with B glycolipids or B antigens on its surface. Now you might have heard of type AB blood. These erythrocytes in type AB blood have both A antigens and B antigens. And I know you've heard of type O blood, if nothing else from the first slide I showed. Type O blood doesn't have either. So not that it doesn't have any antigens, but it doesn't have either of these ABO antigens. So no A and no B. Now let's look at Rh factor. There's many different Rh transmembrane proteins, lots of subtypes, but the one that's used the most often because it's the most immunogenic, can cause an immune response, is the D. So we're just going to deal with this Rh factor. We're going to pretend there's only one Rh factor. And it is a protein, and it is on the surface of red blood cells. Okay, now if you have that protein, that Rh factor on your red blood cells, you are called positive. It would be more appropriate perhaps to say Rh positive, but that isn't what's routinely done. Usually you just say positive. If you don't have the Rh factor on your red blood cells, you're called negative. So that leaves us with 8 different types of blood if you consider both blood groups or blood antigen groups, the ABO combined with the Rh factor. Somebody could be type A blood and either positive or negative, depending on the presence of Rh or the absence of Rh. That's what the negative means. Somebody could be type B blood with the B antigens, and then again, either positive or negative, depending on their Rh factor status. Somebody could be type AB blood and either positive or negative. And someone else could be type O positive or negative. So there's 8 different types. Now, you probably know that these blood types are done, typing is done prior to transfusing a patient with blood. And so we need to talk about the transfusion reactions that can happen. But to do that, we need to consider another chemical, I guess. It's a protein. It's a protein in a person's blood, and these are antibodies. So antibodies vary in different blood types. So we've learned so far that a person's blood type is determined by what antigens are on the red blood cell. So I'm just going to make this up here. This person is Rh positive. So they are A positive blood. This is A positive blood I just drew here. And in the blood, there are white blood cells, there's platelets, but there's also antibodies floating around. And these antibodies are in the plasma. They're kind of Y-shaped molecules. And the tips of the antibodies that are colored blue can bind to antigens and neutralize foreign antigens or tag a foreign cell to be phagocytized. So these antibodies, we don't want to be the same or specific for the antigens in this person's blood because it would destroy their red blood cells. So the antibodies are going to be specific for non-self antigens. Okay, non-self antigens, anything that's foreign. So for example, let's see, there are antibodies against A antigens. So this antibody would bind to an A antigen. This antibody would bind to a B antigen, and this antibody binds to an Rh. Now, what do? I briefly alluded to it. The antibodies can bind antigens on cells, red blood cells included. And when that happens, you get this aggregation of cells. The antibodies can bind to 2 different cells, and you get this big clumping. This is called an agglutination. It's not the same as a clot, okay? So we don't want this to happen in our own blood. This would be bad. Therefore, the antibodies in a person's plasma are different or opposite than the antigens found on their red blood cells. So let's take an example. A negative blood. Okay, let's take that example. I'll first draw a picture. So I'm going to first draw a blood vessel. It's not the best blood vessel you've ever seen, and then an erythrocyte. I'll draw a couple of erythrocytes. This person is A negative, which means the only thing on the red blood cells that we need to be worried about is the A, because negative means there's no Rh. Now, also present in the blood are antibodies. So I'm going to draw some antibodies. Now, these antibodies, we do not want to be specific for A because that would clump up the person's blood. So instead, those antibodies would bind to B or Rh. They always, one antibody only binds to one of the types. Okay, so therefore, if this person was to receive a red blood cell that had a B on it, that would be foreign to them because they're type A, right? So this B antibody would bind to that B. And we'll do that again in a little bit. First, I just want to talk about blood typing and the antigens and antibodies present in a certain type of blood. This is relevant to your first week lab as well. So this person, we've collected their blood and determined that it's A negative. That means the only thing on the red blood cell is an A antigen. In their plasma, there are antibodies against B and antibodies against RH. So no agglutination will occur in their blood vessels, which is good. You don't want agglutination in your own bloodstream. Now there's one caveat to a person who's RH negative, and that is somebody that's RH negative, you would assume that RH is foreign and therefore they have antibodies against it. But that isn't really true. RH negative people don't automatically make antibodies specific for RH unless they've been exposed to RH. And most people haven't been exposed. So we call these individuals non-sensitized. I don't know why we don't just say not exposed, but they've never been exposed to the RH factor. And so they don't make the antibodies yet. But if they ever are exposed to the RH factor, now their bodies will make RH-specific antibodies because sensitization has occurred. And this happens primarily in pregnancy, and we'll talk about that too. So in summary, blood type for blood types. Blood type A, the red blood cells have A antigens. And in the blood plasma are anti-B antibodies. If they haven't been exposed to RH, you won't see RH antibodies. Somebody who has type B blood has B antigens on the red blood cells, and in their plasma, anti-A antibodies. Now let's look at group AB individuals. So people that have AB blood have both types of antigens on the surface of their red blood cells. Therefore, they don't make antibodies against A or B. So this is the universal recipient. If you have type AB blood, you can receive A blood, B blood, or AB blood because you don't have antibodies against those antigens. Somebody who is group O, type O blood, they don't have A's or B's on the surface of their red blood cells. So A and B are both foreign, so they make antibodies against both A and B. Now let's consider this RH business a little bit anyway. If someone is RH positive, they have RH factor on their red blood cells, so they won't make antibodies against RH. And you know, it's not so simple to say the opposite is true for RH negative individuals, because RH negative individuals don't automatically make antibodies unless they've been exposed. So now let's consider transfusion reactions. Okay, some blood type compatibilities. Now, most often what is transfused when a patient needs blood will either be just plasma because they're dehydrated, or they've had hemorrhaging and so they need red blood cells to be able to carry oxygen. So one type of blood that is transfused most commonly are packed red blood cells. No plasma, okay, no plasma, and typically no white blood cells either. So the donor is giving red blood cells, and the donor has antigens on the red blood cells, the donor blood, right? Now, there's no antibodies being given to the person. But if we consider the recipient does have antibodies in the plasma. And so we have to make sure that there's no antibodies in the plasma that match the antigens on the donor. So we have to make sure that compatibility is reached by typing donor's blood and recipient's blood. And we need to know what the antigens are in the donor's blood and the antibodies in the recipient's blood. Now, you can prevent a transfusion reaction by banking your own blood, as well as doing this blood type and only transfusing the right thing. The other thing is you can just always transfuse O negative. And if you transfuse just O negative red blood cells, what you're transfusing is red blood cells that don't have A's, don't have B's, and don't have RH. Therefore, O negative is the universal donor. And I've already mentioned that the universal recipient is somebody who's AB, and I'd go farther and say the universal recipient is somebody who is AB and it's supposed to be a B. My computer's not working. And RH. If they already have RH, then you don't even have to worry if they've been sensitized or not. Okay, it's not drawing very well. Okay, a little bit about the RH sensitization thing. Let's talk about what can happen in certain pregnancies. I don't know how common it is, but there's a disease or disorder called hemolytic disease of the newborn. And what that means is that hemolysis happens at a very fast rate in a newborn. And it has to do with the fact that a mother's antibodies can cross the placenta and get into the bloodstream of the baby. Now, this is due to a complication in RH factor compatibility. Now, there's never a problem with the first pregnancy. So let's say this mother in her first pregnancy is RH negative. And we'll just assume that she's been exposed sometime in her life so that she makes RH antibodies. And now this baby that's developing happens to be RH positive. Normally, there isn't going to be a problem here. Okay, maybe she hasn't been exposed actually. Sorry, she hasn't been exposed. I'm going to do that all over again. We're going to start over. So sorry. And pick a different color because it's not working very good. Pick blue. All right. So this mother is negative for RH. She doesn't make antibodies yet. Her baby is positive, RH positive. When the birth happens, the bloodstream mixes between the baby and the mother. And so now the mom's blood has been exposed to RH factor. And so now she makes antibody against RH. So these are green things are supposed to be RH antibodies. I can't make this work. RH antibodies. Now she has another pregnancy. And again, this child is RH positive. This is a problem because now the antibodies present in the mother's blood can get into the bloodstream of the fetus and cause destruction, agglutination, and destruction of the baby's red blood cells. So what they do is anytime a Rh negative mother has given birth to an Rh positive child and she becomes pregnant again, excuse me, they treat with some kind of a drug. We're going to use this one. This one's trademark is ROGAM. And what ROGAM does is it binds to the red blood cells in the baby. So it basically hides the Rh factor on the fetus's red blood cells. So now the antibodies can't bind to it. Okay? So the ROGAM binds to fetal antigens on their red blood cells. So that's all for the week 1 lectures and blood typing in particular. Thank you for listening.